Hello, I'm Gokul and welcome to the channel. Today we'll be discussing myelination and saltatory conduction. There are a great deal of neurons in the human body, and they certainly don't conduct action potentials at the same rate. Some neurons conduct action potentials very quickly, whereas others do so slowly. So what exactly determines the speed at which they travel? From a physics perspective, action potentials are nothing more than moving electrical currents. The resistance of an electrical current moving in a wire will depend on its internal resistivity, the length of the wire, and the wire's cross-sectional area. This equation on the left, R equals rho L over A, shows that wires that are thicker, meaning that they have a larger cross-sectional area, and shorter will propagate current at a higher rate because the resistance is lower. In an analogous way, if we imagine that the axon of the neuron is the wire and the action potential is the moving current, we can apply the same equation here. An axon with a larger diameter will move action potentials more quickly because a larger diameter means a smaller resistance. Likewise, a shorter axon in the neuron will decrease the resistance of the action potential and increase the speed of electrical propagation. The size and length of the axon is limited by the size of our body. That is, we can't make the axons of the neurons too thick or too short to increase speed of the electrical propagation. Luckily, our body has another way of increasing the speed of conduction. This is by covering parts of the axon with an insulating material called the myelin sheath, which greatly increases the velocity of the action potential in the neuron. Myelin is an insulating layer that surrounds the axons of the neurons. Composed primarily of lipids, myelin helps to prevent action potentials, which are the electrical signals that travel along axons, from decaying due to electrical current leaking out through the axonal membrane. Myelinated axons thus conduct action potentials more quickly and more efficiently than unmyelinated axons. And because of this, many neurons in the nervous system are myelinated. Myelin does not cover an axon fully. Instead, there are intermittent gaps in the myelin where the axon is exposed to extracellular space. These gaps along the axon are called nodes of Ranvier, and the sections of myelin that are adjacent to the nodes of Ranvier are called internodes. The nodes of Ranvier are rich in sodium Na+ channels, which open in response to an action potential traveling down an axon, allowing positive sodium ions to rush inwards. This influx of sodium ions rejuvenates the action potential and helps to prevent it from dying out as it proceeds along the axon. Because the nodes of Ranvier are not myelinated, however, the action potential slows down at each node and speeds up as it travels along the myelinated internode. This gives the appearance that an action potential is jumping from node to node, which is known as saltatory conduction. Myelin is formed by glial cells, but the particular type of glial cell responsible for myelinating axons is different in the peripheral nervous system and the central nervous system. In the peripheral nervous system, glial cells, called Schwann cells, form myelin. Each Schwann cell wraps around one segment of an axon many times to form one internode. In the central nervous system, however, glial cells, called oligodendrocytes, form myelin. One oligodendrocyte can produce dozens of internodes on multiple axons. Because myelin is white, myelinated axons appear white and make up what is known as the white matter of the brain. We can see the importance of myelin to healthy neural tissues and their functioning when we look at diseases where myelination is disrupted. In multiple sclerosis, a neurodegenerative disorder, for example, deficiencies in the myelin sheath can disrupt neuronal communication, resulting in a variety of physical and psychiatric symptoms. That's it for today. Thanks for listening.